Okay, welcome everyone. I am extremely glad to welcome Lisa Manning to our department. She is a tenured professor at Syracuse University. As you can see, she's a young, bright, shining star in the area of jamming and biophysics, so very cutting-edge science at the interface between what physics can do in biology. That's the main topic that we're on today. And she has had so many accolades that I'm not actually going to list them, but I wanted to mention a couple of things about Lisa. So she did her PhD at University of California, Santa Barbara, and then she she started working on the glass problems, looking at random networks and looking at packing and jamming and soft matter physics, things that we're quite familiar with. And then she decided that cracking the glass problem, even though it's one of the most important questions in science, as determined, of course, by the Science Foundation. So she's working on that very important problem with some very prominent collaborators and so forth. But she then decided that that wasn't enough for her and that she wanted to see if there were relationships between cracking the glass problem and some glassy dynamics and physics and relationships between structure and dynamics in biology. So today she's going to tell us more about how physics may have something to say about adaptation in vivo in terms of mechanics. With these? Okay.
Maybe
so
off?
Welcome to Lisa. And then we don't do this.
Okay. All right.
Or you want the three?
Whatever. Down.
Maybe down. Maybe off for 3.
3? I'll try the 3.
That's.
Well, it was 3.
All right. Well, I'll start, and maybe the lighting will continue. Hi, everyone. Thank you for coming. I'm from Syracuse University, not from Columbia, as the flyer misadvertised. So that's even further upstate, I guess. So today I'm going to be talking to you about biology, and I'll explain in a minute what's going on back here. So hold out for that. Before I begin, I want to mention the people with whom I worked, especially a really excellent postdoctoral associate who's starting at Northeastern, another one, Matthias Merkel, folks at Syracuse in the soft matter group, Joseph Case and his group, Eva Maria Schatz-Collins and her group, and the Fredberg group at the Harvard School of Public Health. The main question I want to start you thinking about is, are you a solid or a fluid? So I would say mostly you support shear stresses, right? You're sitting, you can stand. So I would say for the most part you think about yourself as a solid. There's some blood and some other things which are fluid-like. Okay, but you can think that maybe there was a time early on when you weren't so solid-like. So if you think about a developing embryo, actually there has to be very large-scale flows of cells over very long distances in order for the cells to end up in the correct place to form the parts of your body. And actually you may think that at that time it might be useful to have a fluid-like rheology instead of the solid-like one that you need at the end of the day to do what you need to do functionally. All right, so this is some data now from early embryonic zebrafish tissues. So each one of these things is a ball of cells of about 10,000 cells. I'll have you watch this one first in real time. It compresses the ball of cells, and then you can watch it snap back. Not all the way, but part of the way. So on short time scales, that was real time, you can see there's sort of elastic-like spring back. Now this experiment is over a much longer time scale, so it's the same balls of cells, so thousands and thousands of cells in each one of those balls. And now you simply place them together in a petri dish, and what you can see is the cells spontaneously round up from 2 droplets into 1 droplet. And actually, you can make that much more precise. You can look at the scaling of the neck, you can look at the shape, and it's actually very much like 2 oil drops would fuse together in water, for example. And so on longer time scales, this tissue behaves much more like a fluid. All right. So are there other examples of these sort of fluid to solid behaviors in tissues? Here's another one. So this is cultured lung epithelial. So collaborators take cells from the lungs of human patients, either with asthma or without asthma, and culture them at an air-liquid interface. And then you sort of look down from on top like a bird's eye view. And that's what these pictures are. And the days are telling you how long those cultures have been cultured at the air-liquid interface. So hopefully you can see here that there's some cell motion. At day 3, you can sort of see the cells moving past each other and flowing. And then at day 6, you may see a tiny bit of motion, but not much. And then by day 10, very little motion. So there's sort of a transition from cells being very motile to not being motile. And you might say, okay, that's very phenomenological. How are you going to really check and see what's going on? Is this really a solid? Is it really a fluid? So one way to do that is you can look at mean squared displacements of cells, how far they move as a function of time. A neater trick from the glass physics community is to look at so-called 4-point susceptibilities. So you look at correlations between 2 time points and 2 space points as a marker of there being dynamical heterogeneities, which you expect to see in a disordered fluid to solid transition. And you can actually measure those and quantify those from doing some careful work on movies. And you can see very precisely that on day 3, there is dynamical heterogeneities that occur on this time scale of about 80 minutes. And then sort of by day 6, they're sort of off. The dynamical heterogeneities take longer and longer, which means that it's more and more difficult for cells to rearrange. And so this is a very precise measure of whether something is becoming more solid-like. Okay, another thing that one can do is look at cancer cell lines. And so it turns out that these are balls of cells now taken from a breast cancer cell panel. And you can see when I run these movies that some of them fuse together, very much like a fluid. This case is really interesting. They get stuck in a dumbbell shape, like a figure 8 shape, and then these don't really fuse together very much at all. And so again, you can see there's a transition across the fluid-solid transition evident in this breast cancer panel cell line. This is behaving like a fluid, and this is behaving like a solid. Okay, and so again, we can quantify that. I'm not going to show the data here. What I'd like to ask you is, can we understand what controls these fluid-solid transitions in tissues? Because it really may be important for biological function and for disease. And so the obvious thing that one might think to look at, especially since I showed you data from sort of measuring these things using glass physics, is let me look at what we know from jamming and glass physics, which has been developed, in some cases here at NYU over the past 20 years. Right? And so we can ask, well, what happens when you have strongly interacting objects at very high densities? So this is a picture of cells at a high density. These are nuclei in blue and membrane marker in green. And actually it looks, you might think that this is very different from, say, mayonnaise. Okay, mayonnaise is a good example of a material that becomes jammed and turns into a solid. So how many of you have made mayonnaise before or seen someone here make mayonnaise? Okay, and so what do? You start out with 2 liquids, right? An oil and water, and then you have some sort of emulsifier, and you mix droplets of oil into your water or water base until you get enough of those droplets so they start packing together and packing together. And once they're tightly packed enough, you get something that can stand up to its own weight, that has a yield stress. That's a solid. And so changing the density is a good way to get something from being a fluid and a solid. And you can see these are also at very high density. All right. So this is a picture of what it looks like inside a dense emulsion, for example. And you can see there's some similarities, but there are some obvious, very big differences. Right? So that's what we're going to be talking about for the rest of the time in this talk today. All right. By the way, this isn't just mayonnaise. There's all sorts of materials that have this sort of behavior, including earthquake faults and bulk metallic glasses and things like that. And so people want to put these things on a phase diagram to understand for these non-biological materials, what parameters do I have to change in order to get them to solidify, to go from being unjammed to jammed. And this jamming phase diagram is a way that people tend to organize data in this field. And so on this axis is 1 over the density. Oh, so let me first say this axis. Okay, so what happens if I take a material, and like water, and I cool it down. I get ice as a solid, right? That's a crystalline transition. But if the material stays disordered as it solidifies, that's called a glass transition. So that's this axis. And the green is the solid or jammed region, and the blue is unjammed. Okay? Another thing I can do is change the density. That's on this axis. So if I have a low density, then I'm more fluid-like. And as I compress the objects together, I go through a jamming transition and I can become a solid that way. Right? So I'll be coming back to these two terms later in the talk in the context of biology. So I just wanted to fix notation. And so this is the sort of picture we have in mind there. All right, so another thing you can say is, well, one obvious difference between cells and those foams that you were showing me is that there's adhesion. So what happens to this jamming phase diagram when I have sticky particles? And there's been work done on that, and it turns out that you can basically replace this temperature axis by something that looks like temperature over adhesion. In other words, if I make my particles more sticky, they're more likely to jam. That's the result, fairly intuitive. Okay, so how do I know, or how do I quantify that a material is undergoing this type of transition? So one of the ways I can do it, as I was saying earlier, is by looking at the mean squared displacement. So that's, how far one of these particles moves as a function of time. And you can see in many of these viscoelastic systems, you see sort of plateau where particles are caged by their neighbors, so they don't move as far as you think they should in a given time, and a crossover to a fluid-like behavior. So this is a log-log plot, and so if the displacement goes like the square root of time, then I expect the squared displacement to go linearly in time. So this would be slope 1 over here, and that's what you can look for a long-term diffusive behavior. Yes?
That's just ordinary diffusion.
This is just, so this is I take a colloidal glass like this, and I track the particles, and I look to see what they're doing. And because they're dense, so this isn't pure diffusion. If it was pure diffusion, I might have a ballistic regime that's sort of, before they know that they're sort of diffusing around, crossed over to a diffusive regime. But in these dense systems, I get this extra plateau. Which
I was just asking about the straight lines that you draw.
Oh yeah, this
Straight
is
lines are diffused.
this is diffusive, and I think this is steeper. It's ballistic. Okay, thanks. Great. All right, so another way that you can check and see if this sort of caging or crowding behavior is responsible for a fluid-solid transition is I can look at the shapes of trajectories. Okay, and so if, let's say, I'm jiggling around in a cage of my neighbors, then it looks something like this, a cloud of Gaussian points. And then every once in a while, I can break past my neighbors. I sort of jump to a new set of neighbors. And so that looks a little like this. It's a little cloud and then a jump and a little cloud. Okay, and then finally, after a long time, I've done a lot of those jumps, and it begins to look Gaussian again. So one way to sort of look for this caging behavior in trajectories is you look for where this trajectory looks the most non-Gaussian. And that's what's plotted here. This is how non-Gaussian a trajectory looks as a function of time. And you see a peak right here, and that happens at the cage, what's called the cage breaking time scale. Okay, so that's the idea. And that's another way to tell that you have this sort of behavior going on. And you can ask, well, does this really happen in biology? When you were sort of saying there's these fluid-solid transitions. So this is some data from a zebrafish embryo. These are a couple sample trajectories where you can sort of see a cell is jiggling around and then makes a jump and jiggles around. And to get statistically significant, we look at a whole lot of trajectories within the 3 dimensions in one of these embryos. And again, you see a peak in the non-Gaussian parameter here that roughly coincides with the onset, sort of the transition from subdiffusive to diffusive behavior in these systems. And you can even look at a video. So this is some images of nuclei that are stained inside one of these zebrafish embryos. So look at the one with the arrow on it. And you can see it sort of jiggles around and then ducks under the other ones and finds a new set of neighbors. So you sort of have the same set of neighbors for a while and then jump. Okay, so why am I, sure.
In contrast to what you had before, this is biology of living systems before you had cultures.
Yeah, so there's several systems. This is actually called ex vivo because it's an explant. So it's a whole zebrafish embryo, but it's been sort of popped off of its yolk so that you can image it this way. But it's sort of, you know, it's still the same set of cells that was in the embryo. So it's sort of a mixture between an in vitro and an in vivo system. Great. Okay, so why am I going to all the trouble to check and see if this really looks like a glass transition? Well, because maybe then I could go and see if I could predict, based on a jamming phase diagram, what cells should do to go back and forth across this transition. Right? So this is, again, the same picture of these epithelial cells, and then this chi4, I just wanted to show this to you here again. This chi4 is also sort of, it's the time scale over which I see dynamical heterogeneities, and in most glasses that's consistent sort of with a cage breaking time scale as well. Okay, so this is all a bunch of data showing you that yes, there's glassy dynamics experimentally observed in tissues, and now we need an explanation for it. Okay, so how could we model this? Well, one way we could model this is by making a packing of particles in a simulation that are self-propelled. And we could make them very, very dense. And you could ask what happens to an active self-propelled particle system when I compress it, right? And so there's lots of people that have done this, including me, and some really interesting non-equilibrium behavior comes out of it, including some interesting phase separation. There's a lot of really neat new physics in here. I won't have too much time to talk about that today, but I'd be happy to talk to you about it later. The important result for the glass transition is that it's nearly identical to that for adhesive colloids. So again, remember the glass transition, if instead of temperature up here on this axis, I replace temperature by particle speed in these self-propelled particle models, then I can still think about this axis as the glass transition. And if I do that, it looks very, very similar, with one little asterisk. And the one little asterisk is that there's an additional parameter, which is for these self-propelled particle models, which is how long does one of these particles move in a straight line before it turns? And that's the persistence length. And so you can ask, well, in addition to the speed, what's the effect of the persistence length? And so this is the particle speed on this axis. This is changing the density. And so this is a phase transition line measured in simulations. And so over here is liquid state, and over here is a solid state. And then you can ask what happens. So this is in the limit of vanishing persistence. So this is sort of what colloids look like because they don't have any persistence. All right, and then as I increase the persistence, the interesting result is I actually shift this phase transition line a little bit. It's a small amount. You have to look carefully for it, but there's some interesting physics there as well. So there's some difference in how you approach ground states of a complex potential energy landscape depending on how persistent motion is. Okay, but the point is that this is sort of a very small effect. This, I've drawn it big here. It's actually very small. It's like a hundredth of a sort of 1/100th of an effect on this number. So it's in the second digit.
What is particle speed for colloids?
So it's basically, the particle speed squared is the kinetic energy for colloids. So the temperature sets the particle speed in a colloid.
Oh, these are not active colloidal particles.
What?
These are not active colloidal particles.
No, so no, this is just the sort of the...
Back to temperature.
So this is sort of, if you take the limit of this model so that the fluctuations look like thermal fluctuations, that's what I'm calling colloids here. I know a lot of people at NYU work on active colloids. These are passive colloids. Okay, and this is going in the direction of active colloids. So as I make the active colloid go, have a persistence length, so it goes in a straight line for a while because it's being propelled. You're going along that direction. Thank you. Yeah. All right. So given that these active colloids are very much like the old jamming phase diagram, some bioengineers sort of conjectured about 3 years ago now that this jamming phase diagram could be imported wholesale to explain the behavior of tissues. So they published a paper and they proposed this phase diagram, and it might look a little familiar because it's exactly the same as this phase diagram except they just got rid of temperature, okay? So it's the same. Okay, and it's a conjecture, but I think an interesting one. They said maybe we can organize data about biology since we know that these adhesive, these active particles are so much like the passive particles. Maybe we can do this. Okay, and so when I saw this paper, I liked it, but I was frustrated. And the reason I was frustrated is because if I look back at this diagram, a lot of the behavior of this phase diagram is governed by the critical point that happens as a function of density. In other words, changes in density are what drive, at the end of the day, the glass transition in particulate matter. Okay? And in confluent tissues, where there's no gaps between cells, all the cells are touching one another. So this is a confocal slice through a three-dimensional tissue. So you're looking at a two-dimensional slice through a three-dimensional zebrafish tissue. And you can see that in this case, there's no changing the density, right? This is at packing fraction 1. So nevertheless, this undergoes a fluid-solid transition. So we have no theory for that. What do we do? So that's what I'm going to talk about for the next little bit.
What was this motility? Was that related to this length?
What motility? This. This is what they sort of said was the speed. So they said this motility should be the speed of the particle.
And where is this persistence?
So in this phase diagram, it doesn't show up. So we know you might imagine it should make a difference. And actually, hopefully by the end of this talk, I will show you a phase diagram that has that as a parameter. Okay, it should be there. That's another problem. All right. Again, they didn't try to make that figure precise anyway. They sort of waved their hands and said it was maybe a way to organize data. Okay, so now what are we going to do? We're going to say, okay, a jamming transition governed by density changes is impossible. Is there an analog to the jamming rigidity transition in confluent tissues? So to answer that question, we're going to look at a particular model, which is called the Vertex model for tissues. And it was developed about 15 years ago. And it was developed in order to make predictions about cell shapes in sort of developmental biology systems. Especially, it works really well in fruit fly embryos. Okay, and so they tried to make predictions about like the number of sides that we expect a cell to have. That's what this model was used for. Okay, and so it basically represents, instead of representing cells as squishy spheres, it represents the edges and vertices between cells. Those are the objects. And so then you write down an energy functional that you think might be relevant for explaining how much work does it take for a cell to have a certain shape. That's the question we're going to be asking. Okay, and this is what people write down. For this audience, I'm not going to go into too many of the details, but let me tell you what it is. So this is, again, looking at a bird's eye view of a monolayer of cells. So it's a single layer of cells, and I'm looking at the outlines. And one of the obvious things I can measure is the perimeter, and I'll call that P. And another obvious thing I can measure in a picture like this is the cross-sectional area, A, of one of these monolayers. Okay, and so then what this equation says is it says that cells have a target area that they want to maintain and a target perimeter that they want to maintain. And then this is, for physicists, this is the simplest energy functional you can write down to second order, basically, that says that the area sort of is quadratically coupled to this target area, and same for the perimeter. And this line down here tells you why it's reasonable biologically. So for example, this term comes from the fact that the cells in these monolayers are essentially incompressible in 3 dimensions. And so because that and height fluctuations are reduced by adhesion molecules, you get something like this. This is due to some motors and biopolymers inside the cell. And this term is due to some interfacial tension and adhesion. And all of these terms are backed out, or sort of backed up by specific measurements taken on single cells, on groups of cells, using micropipette aspiration, using laser ablation, and things like this. But the nice thing is for a physicist, it can be written this way, and this seems tractable. Right? So then one can non-dimensionalize this model, and you basically find that there's 2 parameters. This one is the most important, so I'm going to take a second to explain it. It's called the target shape index. So it's this target perimeter that a cell wants to have divided by the square root of the target area. Okay? And so that's sort of how sort of elongated a cell wants to be, right? Because if this number is small, then you want to be a sphere, right? And if this number is large, you want to sort of be elongated or star-shaped or something. Okay. So this is an important parameter. This is the ratio between these two stiffnesses, and that turns out that parameter is less interesting. Okay, there's a third one that I'm not going to talk about today. All right. So rearrangements and migration. So this model had been around for 15 years. What we wanted to do was understand how it might be used to understand the dynamics in a tissue. So if I want to understand how quickly a cell migrates through one of these two-dimensional tissues, the first point, which is probably obvious to you in the audience, the only thing that can happen is that I have a series of T1 transitions. So what's a T1 transition? It's when two cells that initially aren't touching, they lose sort of this edge shrinks, and they become neighbors, and then here they're neighbors, and the two cells that were formerly touching are now not neighbors. And this movie that I am going to try to show you one more time, you might think that it's a movie of a simulation of the cell crawling, but it's just literally a series of T1 transitions around that cell to demonstrate that you can migrate via just this mechanism. And in fact, that's the only game in town. Okay, so then what one can do in the framework of a simulation is ask, well, what's the energy barrier to one of these T1 transitions? I have an energy functional. I can ask about energy barriers in this landscape. And so what we can do is we can test every single T1 transition in one of these simulated tissues and ask how much energy does it take to go through that entire process. And then this is what we denote the energy barrier. Okay, and then I can ask, well, what are the statistics of the energy barriers? So I'm not going to talk too much about the statistics today, although they're kind of interesting. I'm going to talk about the average. So this is the average energy barrier in a tissue as a function of those two model parameters I told you about. One is this P0. This is this target shape index I was telling you about, this perimeter to area ratio. And then these are at different values of R. And when I first saw this, I was very surprised because, sure.
This is an average. You take a given configuration and you average over all the cells in that configuration.
And we also do it for different configurations as well.
And average over configuration.
Yes, both. We average over both. It turns out, and we've
dynamics at this stage.
No dynamics at this stage, except we're just enforcing a particular path through phase space for a T1 transition and asking how much that energy cost is. Right, that's exactly right. And so the surprising thing to me about this image is this plot, is that on this side here, when this P0 value is greater than about 3.8, there's a finite energy barrier to rearrangements. But if I'm on this side, all of them have essentially zero energy barrier, which means that cells can basically move around for free on this side. And that was very unexpected. And so when you see something like that, then the next thing you might ask is, well, this sort of looks like a rigidity transition. What kind of phase transition is it? Is it a first order transition? Is it a second order transition? Okay, and so what you might think to do then is come up with a scaling ansatz and try to collapse this data. So there's plenty of examples of this approach for these sort of second order phase transitions, rigidity transitions. A common one is for granular particles. Another one is for rigidity transitions. So I don't expect you to, this is a busy slide. I don't want you to follow all the details. What I want you to see is that by having a critical sort of, you have to understand what's controlling the critical point. So in jammed particles, what's controlling the critical point is the packing fraction or the density. So phi minus phi j, the distance to the critical point. And then there's a top branch here, which is a fluid, and a bottom branch, which is a solid, depending on which side of the transition you are. Okay, and you can extract scaling exponents for this. And you see a similar thing for random networks, and the control parameter is the bond occupation probability. Okay, in our case, the control parameter should be this target shape index. So let's do that. We just come up with a scaling ansatz and see if the data collapses. And it collapses beautifully. Okay, and so what you see here is this is going to be the branch where we have a finite energy barrier. So this is a rescaled energy barrier. And on this branch, it's finite. And that's when our target shape index is less than this critical value. And over here, we have no energy barriers, and that's when this target shape index is greater than its critical value. We can extract exponents, which look rather mean field, and perhaps more importantly, for biologists, we can also pinpoint the exact location of this transition by seeing when the scaling collapses best. Right? So this is pretty neat. This is just a model that's been in the literature for 15 years, and it had this beautiful second-order phase transition embedded in it. All right. So the next thing we can ask is, what does this mean for the sort of jamming phase diagram? Okay, so for particulate matter, if I want to make my particles turn into a solid, do I make them more or less sticky? So I want a solid. Do I want to make my particles more or less sticky? More sticky. Okay, I know, I'm sorry, you know this already. Okay, particulate matter, the axis is 1 over adhesion because I want my particles to be more solid-like. I want them to gel. But interestingly, in this model for biological tissues, the axis is opposite. And that's a little perhaps surprising. But the reason is simple. In confluent tissues, when I have more adhesion, that means I want to have more surface area in contact with other cells, right? Because I get benefit from adhering to them. And in one of these models where the degrees of freedom are the edges and the vertices, having a larger perimeter or a larger surface area in 3D means I actually have more degrees of freedom, more ways to move those edges and vertices. So in fact, in this model, we have more degrees of freedom and more liquid-like behavior with more adhesion, which is definitely counterintuitive. All right, another interesting prediction of this model is that there is an order parameter, a structural order parameter. And to me, as a person that's worked on glass physics for a long time, that's very exciting because in glass physics, for particulate matter, there is no structural order parameter. But here there is, so let me tell you about it. Okay, let's define an observable shape index. So recall that this P0 is a model parameter. It's the perimeter to area ratio the cells want to have, which is governed by things like the number of adhesion molecules expressed and how stiff the cells are. Okay? But I can ask, in an actual simulation or an actual experiment, do cells get to actually meet the target? So this is the target, but now I can measure what they actually do. So this is the perimeter, the actual perimeter divided by the square root of the actual area. And I can ask, what happens? So if I'm on the fluid-like side of the transition here, I see that the sort of observed perimeter to area ratio matches the target perimeter to area ratio, which is, this is just a straight line. So on the fluid side, the cells can meet their target. On the other hand, on the solid side, over here, that's this side, it's pinned. It is pinned right at this number, 3.81, okay? So that's pretty exciting to a biologist because this prediction here is a jammed tissue should have this observable that is precisely 3.81. And as I become more and more unjammed, then I should go away from that. And so no longer do I have to do, if this is correct, I don't have to do a lot of work to measure mean squared displacements or dynamical quantities. I can just look at a snapshot of cells and tell you whether that tissue is a fluid or a solid. All right? And so I sort of suggested this to Jeff Fredberg, who had all of this nice data on these cultured lung epithelia. And he already had very precise data on which of these were jammed by looking at these chi-4 numbers. So in other words, we have a really, I'm not going to go through all the details here, but we have a really good handle on which of these tissues is precisely jammed using dynamical data. All right? And so we can look at non-asthma patients and asthma patients, and using some of those metrics I showed you, we can say with certainty that this is jammed on this day, and it's almost jammed here, and almost jammed here. And this is this cell shape index that I was talking about. So what's our prediction? Our prediction for this data is that it should be sort of pinned here when it's jammed and rise away as it becomes unjammed, right? That's our prediction, that this number should just sort of be here and rise away. All right. And that's what the actual data looks like. So it's really at this number which we predicted a priori from pure theory with no fit parameters, right? So this is solid, right? And it's fluid, and this number gets larger as you move away. So that's pretty neat. Okay, so.
But the error bars include no effect.
Oh, so actually, that's a great question. So these error bars are huge, right? Absolutely huge. And there's several sources for them. Number one is that you see these fluctuations at this level in the simulations as well. There are inherent fluctuations in this. Although they get, I could go into a lot more detail. We think that there's a contribution of this to these fluctuations due to disorder in the system, and there's also a contribution due to motility. Because in the limit, right as you get to the transition, actually, interestingly, the fluctuations in this way go to 0, which is the opposite of many. So anyway, we understand a lot about the fluctuations, and they should be there. We expect them to be there.
What's the meaning of this rectangle if you have these huge error bars?
Yeah, so this is sort of the, I think 75% of the data falls in this rectangle, and this is including all the outliers. And the bar here is the median. And so again, when you plot the same data for simulations, what you'll see is the order of magnitude of these curves is actually very similar, but on the day, sort of in the simulations where it's jammed, the error bar actually goes to 0 unless you add active motility, which is what I'm about to talk about next. So for a model where there's no active fluctuations, there should be error bars that are about this large for this one and about that large for this one, but not very big for this one. Okay? That's the idea. Okay, so to sort of get towards that point, when I was talking to Jeff Fredberg about this, he says, okay, Lisa, fine, it sort of worked, but real cells are moving around actively. We can see them, right? They're pulling on their neighbors, they're pulling on their substrate. There was absolutely no activity in your model whatsoever, right? What is the effect of having a given persistence for the persistence that a cell wants to walk? What does its change in speed do? So what we can do is we can add that to our model. And we want to do so in such a way that we can simulate this model in both 2 dimensions and 3 dimensions. And the way we do it is by using a Voronoi tessellation and imbuing that tessellation with this energy functional. And then we add two parameters. We add sort of persistence, and so we have particles that move at a velocity, at a speed v0, and a direction given by theta, and that theta has a noise associated with it. So this is just the magnitude of the white noise and the fluctuations in the angle. So the bottom line is there's a parameter for the speed and a parameter for the persistence. That's V0 and dr. And otherwise, the interactions between the cells are as close as we can get them to the Vertex model I showed you before, although not exactly. And there's this sort of effective multi-body interaction here. Okay, so now what we can do is I'm going to show you a phase diagram which just has one of those axes. This is the speed of cells now. And this is your old familiar parameter, which is the shape parameter. And so this is sort of cell mechanics if you want, and this is cell speed. And so the first thing you would want to check is that at 0 speed, do I still see the transition between solid and fluid right at 3.81? And I do. Okay? But now as I turn on speed, as I turn on cell speed, I go from being a solid to a fluid, right? So I sit here and then I turn on cell speed. And I can check, and so these dots here are dynamical data taken from sort of based on mean squared displacement data to see whether trajectories are diffusive, like this in the fluid, or non-diffusive, like this in the solid. Okay, and so you can even look at movies. They're kind of mesmerizing. They don't tell you that much, but they're kind of cute. Okay, and so the other thing you could ask is, well, you claimed that there was a structural order parameter for that transition. And so does it still work now that you've added all of these fluctuations in? So what I'm going to show you now is this same plot. So again, this is the transition at 3.81, and this is a line, if you will, of glass transitions that are governed by this critical point down here. And I can say, what is the shape order parameter? So what I'm going to do is I'm going to draw a line on this graph. So this is dynamical data taken from whether cells are diffusing or not. And now I'm going to put on top of it a structural piece of data, which is the line at which the shape order parameter goes from being below 3.81 to being above 3.81. And I'm just going to draw it on top of this plot. And it's right there. So I just didn't draw a line. You know, this is a different set of data. So this is dynamical data, and this is structural data, and you see they match up basically perfectly. So this is a sort of interesting thing. It means that the structural order parameter survives the fluctuations due to activity. And it also survives what happens when I add persistence. So I can sort of ask, what happens when I make my cell tracks more persistent? So let me take a system that's over here, and it's a solid. So this white stuff is a solid, and this gray stuff is a fluid. And what happens if I make it more persistent, which is going this way? You can see that I can fluidize one of these tissues just by making cells more persistent. So this is a new phase diagram from an actual model. It's not a, I mean, it's still a model, but it's not a conjecture as the previous one, that tells you how you should change cell-cell mechanical properties like adhesion or cortical tension, or the speed, or the persistence of cells to go from a fluid to a solid. And the suggestion is that biologists could begin to organize their data for signaling pathways that alter these things in this way along these axes, and it could be very useful for understanding why some tissues behave collectively the way they do. Because this is something that you would never get from a single cell analysis, right? This is something that can only come about collectively. So it's kind of, yes?
So you showed earlier some tumor tissues where you showed also the different phase transitions. So what are the different for those tissues when you mentioned one tumor and healthy one?
That's a fantastic question. So we don't, so it could be any one of these things, right? This phase diagram says that it could be any of these. And what we're going through really carefully right now is testing persistence. Right now, we're sort of testing the persistence of these cells, testing their speed in isolation, testing sort of, because you can test their mechanical properties with an optical stretcher. So we're sending those through an optical stretcher to get an idea of the statistics. I may show you a tiny bit of that data in a bit. So we don't know the answer, but we're literally doing that right now. All right. Literally, that's the next thing. So in the few minutes I have left, we're looking at primary tumors taken from cancer patients. And the reason you have to use primary tumors is because we know that the properties of cancer cells change when you passage them. So it's really important to get them right off the patient so that you understand what the actual properties are. And we send them through one of these devices called an optical stretcher. And this is what the curves look like. So when you stretch them, you see that the cells either deform more or less under the same power. And so that tells you that the cells are either stiffer or softer, right? And then you can come up with histograms like this that says, what's the relative deformation of a whole population of cells now from either a breast cancer patient or a control patient. And what you see sort of right off the bat is that there's a broad distribution of stiffnesses in these tissues. And this can be sort of, this stiffness or this relative deformation can be mapped to stiffness, which then can be mapped onto this P0 variable, this target shape index variable. And so we can see that there's a broad distribution, and maybe that the breast cancer cells are a little softer. Certainly the distribution is even broader for the cervical cancer. And so in the framework of our model, we can ask questions like, what happens to the rigidity transition when you have a broad distribution of stiffnesses, a broad distribution of P0s? Okay, and so this was the critical rigidity transition that we had before at 3.81. And so now what I'm going to show you is some data. If now instead of all of the cells having the same P0, they now have a Gaussian distributed P0 with different widths. And what you can see is that this this transition sort of gets smoothed out a bit. That's not so surprising. What's kind of interesting is there's a really neat way to process this data. And so what you can conjecture, in fact what my postdoc Max conjectured, is that even though that 3.81 number was for a collection of tissues, he said, well, maybe I can use it at a single cell level. Maybe I can say that if one cell has a P0 value that's less than 3.81, it's a solid, and otherwise it's a fluid. So I can take a mixture of cell stiffnesses that's broadly distributed and make them into a binary group of either soft or stiff cells. And the reason why I would do that is maybe I can write things down in terms of the fraction of cells that are stiff. And the reason I might think to do that is if I think that there's an underlying percolation transition, then that should collapse the data, right? All right, so this is what the data looks like if I plot the fraction of all of that data. If I plot the fraction of rigid cells categorized in this way, you can see it collapses all the data nicely. And that there's a generalized rigidity transition, which we can show is actually a percolation transition that happens here. So sort of that's neat from a physics perspective. From a biology perspective, it's also interesting because it means that a tumor can remain rigid even if over 50% of its cells are soft because of the way it organizes, because there's a percolation transition. And that's important because cancer, it's really, for cancer cells to proliferate, they actually need to proliferate in a stiff environment. And so this is sort of bad because it means that when you have a heterogeneous distribution of stiffnesses, you get cells that can divide because the tumor is still rigid, but the soft ones are sort of the dangerous ones that can move. And I'll show you another way of looking at that. So when you sort of look at, this is sort of biology data. It's difficult if you're not sort of used to looking at it, but you can sort of, what they say in these manuscripts is that cells leave primary tumors. The bad cells leave primary tumors in sort of quasi one-dimensional streams. That's an experimental observation that's been around for a long time. Those are the things that are bad, those cells leaving the tumor. And you can ask, well, what happens to the soft cells? If I just plunk down a heterogeneous mix of cells with different stiffnesses, what happens to the soft cells? And they actually totally move in quasi one-dimensional streams, just spontaneously. You can even watch this in a movie. So this is quantifying that effect, but this movie is worth 1,000 words. So this in this movie, so you could see the effect more easily, I've put all of the soft cells in the middle, and there's some stiff cells around them that you can't see. And then I'm just going to start the movie with activity. And you can see spontaneously that there's that one-dimensional row of cells that spontaneously exited, right? So it starts out as a compact cluster. Oh, it's not going to let me replay it, is it? All right, one more. You see how that sort of, that just spontaneously happens with you doing nothing. So that's pretty interesting that it suggests that the environment of a tumor could possibly play a role in generating these sort of multidimensional streams that can then go out and start new tumors. All right, so I won't talk about 3D. We can do this model in 3D. It has the same behavior. It has the same phase transition. There it is being a fluid. You can measure it. You can measure the transition point. It still has a structural order parameter. So I sort of want to mention that there's a lot, I mean, this is sort of like Candyland for physicists, actually, this model. It's really interesting. There's a ton more to do. So on the physics side, we're working right now on understanding whether this rigidity transition is actually topologically protected and if there's an index theorem associated with it. If those of you who know the Kane-Levinsky paper along those lines, we can see, we believe that there should be fluid hyperuniform states that could potentially have photonic band gaps if you could make a system like this. Maybe, you know, if you could correctly decorate the cells that are in the fluid state, they would also be hyperuniform. All right, we can ask, can pressure, stress, and surface tension be generalized to these systems? Is there a meaningful form of those in these types? On the biology side, we can see if this works in lots of different tissues. So far, it's worked in a lot of them. What's the relationship between this epithelial to mesenchymal transition seen in cancer and this fluid to solid transition that we see in this mechanical model? I think there could be a link. And then we can ask sort of what happens when we prevent T1 transitions actively, because some cells have mechanisms for doing that, and then that generates some interesting mechanical properties too. Okay. So many biological tissues are apparently close to a glass or a jamming transition. We have this Vertex model, which has been around for about 20 years now, but it exhibits a novel type of rigidity transition, and the control parameter is this target shape index. It has a structural order parameter, unlike some glass transitions, which makes it quite nice. We have an active S version of the model that includes speed and persistence, and we can quantify those effects on the behavior of a tissue. In heterogeneous tissues, the rigidity is controlled by the fraction of soft cells, and we can do it in 3D too. All right, so thanks so much for your time. Yeah.
In those, you had some movies near the start where over time a two-dimensional set of cells became rigid.
Yes.
Do you know what's driving that transition? Which one of those parameters in that set of cells is changing?
We are looking at it right now using traction force microscopy. I might have included some of that data sort of as an extra slide. I don't know if I did or not, though. Let's see. Oh, yeah, here it is. So you can look at traction force maps. We certainly know that there's a difference in sort of speed or motility among the two, like sort of the speed that the cells want to move between the asthma, because if you take one of those cells out and like let it try to move by itself, the ones at day 3 move faster than the ones at day 10. But that's not the only thing that's going on. This data sort of suggests that there also might be a change in the adhesive interactions between them, because this is a complicated data set. But anyway, we're looking at adhesive interactions too, and we think adhesion might be changing between them in the way predicted by the model. Yes?
I was really intrigued about what you said about the fluctuations going to 0 at the transition.
Yeah.
So can you say a bit more about that?
Yeah, it's really, and it has to do with our ideas about this index theorem and whether this is topologically protected too. So the thing is that the potential energy landscape on the fluid side of this transition is really interesting. Basically, these cells can move with 0 energy cost, and so and that's because there is sort of always it appears that there's a continuous way to change all of the edges and vertices in the model, so that you always maintain that both the area is exactly at the area it wants to be and the perimeter is exactly at the perimeter it wants to be. So if you can, so that's the point is that even in a disordered system, all the cells can have exactly the perimeter and exactly the area they want to have. So in the absence of like active fluctuations, the disorder doesn't introduce any additional fluctuations, and so the structural fluctuations go to 0 at the transition point.
I think maybe you already answered this. Just put over it. So topologically protected, you mean what is the index?
So I'm talking about like thinking about the Calodine index theorem, which says sort of that the number, it's basically glorified constraint counting, right? And it says that you have a rigidity transition essentially when the number of constraints equals the number of degrees of freedom. I'm talking about that index theorem. And because if you're sort of, that would be a sort of, one way of thinking about that is it's sort of topological, like even if you add higher order terms to this energy functional, that should still tell you when the rigidity transition happens. But it's sort of interesting that just as a comment, if the way that people typically talk about 0 modes in these types of models is by asking whether sort of individual bonds or springs are changing length. And there's something really interesting in this problem where the sort of modes that go to 0 in the Calodine index theorem and the ones that like Kane and Lubinsky used are different from the 0 modes in the dynamical matrix in interesting ways. And so there may not, unlike many other systems, that index theorem may not tell you about the rigidity transition in the system, we think. Does that sort of, I mean, I know that's not a complete answer, but that's the idea.
So this critical value of the shape parameter, 3.81, appeared a bunch of times. Do you have any, is there any like sort of geometrical?
Yeah. So we have a conjecture in our Nature Physics paper, which is complete BS. And the reason I know it's wrong now is because we've done it in 3D and it doesn't work at all in 3D. So that's why it's good to do higher dimensions, right? Now there's another explanation which might be correct still. So we sort of had 2 potential explanations for this 3.81. And it would, I mean, if we understand what's going on with this index theorem as well, that also may shed some light on it. But there's sort of this, let's see if I can bring up, I had a slide just to show you quickly. Yeah, this. So this I think still could be right, way of thinking about it. So the idea is that in 2 dimensions, the topological Gauss-Bonnet theorem tells you that each cell has to have 6 nearest neighbors on average. So we'll have a mean field model where we have hexagons. Okay, and the reason we have hexagons is because we know that has to be correct in mean field. Okay, and then you can ask, well, how much energy is required? Let me keep all of the other edges fixed, and let me just execute a single T1 in the middle of this mean field model, right? And let me ask about the energy cost there. And not surprisingly, in hindsight, you basically have a very slightly perturbed pentagon at the transition. So you have 2 pentagons and 2 septagons, but the pentagons always cost more energy. The sides with fewer, the shapes with fewer sides always cost more energy in this energy functional. And so you can see precisely at 3.81, which happens to be the perimeter to area ratio of a regular pentagon, you allow those pentagons for free. So that energy barrier goes to 0. So that tells you why the energy barriers go to 0, but the curvature goes to 0 at the same place, and this does not explain that. So it's a really, I think it's an interesting, I think there's some fun math to be done here, actually.
Any further questions? So we have, as usual, the wine and the cheese upstairs on the 6th floor. So let's thank Lisa for